Thank you for inviting me. So my name is Jay Konieczka. I'm one of the co-founders of enEvolv, along with Farron Isaacs and George Church, who some of you may know, and also our CEO, Marco Farshid. And we engineer microbes to produce bio-based products, both together with commercial partners and also our own proprietary targets. Our approach is fundamentally different from others in our space. We have technologies inside the cell that allow us to build and screen up to billions of genomic designs in a matter of a month. And I'll tell you a bit about how we do that. And this is also, I should note, without heavy investment in automation. So the challenge in engineering microbes is basically the complexity of biology. So, we're all familiar with the design, build, test, and now analyze and learn, because we don't infringe on trademarks at enEvolv. We insist that we have to analyze them first. So we're all familiar with this, and we all know that, given the complexity of biology, as you iterate through this process, this serial process, you have to be very aware now. We're beyond the scope of just optimizing a simple pathway. We have to be looking at the whole organism, right? So it requires just a tremendous amount of throughput to make it through this process efficiently and explore the vastness of the genome space that we're dealing with. And our platform does exactly that. So this is it in a nutshell. We have variation technology, which delivers mutations in a massively parallel fashion, and I'll tell you about that. And then we screen the cells on an individual cell-by-cell basis in an ultra-high throughput fashion with technology, again, inside the cell. And then, of course, we analyze, characterize, learn from them, and that informs the next design cycle. So the variation technology is actually MAGE. I think most of you are familiar with MAGE. And what MAGE allows us to do is deliver in a massively parallel fashion single point mutations, arrays of point mutations, insertions, deletions at multiple loci in the genome simultaneously. And we do this in a cyclical fashion so that we end up with a culture where every strain is precisely engineered and uniquely different from every other strain in the culture. So there's a combinatorial pool of strain variants, which then poses a new challenge, which is, how do you screen through that genomic haystack to find the good needles? And for that we have sensors, and these are allosteric transcription factors that we actually engineer at enEvolv to recognize the specific and desired target molecule that we want to produce. I don't have time to get into how this works, but in a matter of months to now, in some cases weeks, we can turn around a sensor, and it's actually a panel of sensors that respond in different fashions to the production of the desired target molecule. So these are there and present and driving gene expression, either a GFP or some other kind of reporter, maybe a selection marker. And those strains that are producing more of the target molecule, we can then literally see within the population. So our sweet spot is right there in this cycle, is right there at the build and test phases. If you were going to try and do this with automation, heavy investment in automation, you're going to end up with something like 30 robots, and $10 million in CapEx to just get to 10,000. And these numbers are based off of literature and our own experience in other facilities in our space to do a whole build and test cycle. And this is, roughly within 50% of reality. But for us, the same amount of people babysitting such a robot farm can be running one project with 1 billion strains that we can build and screen. And of course, this scales linearly. If you were to try to do this with traditional automation, it gets a little ridiculous. You're going to need a lab the size of Boston full of robots. So our tech is portable. We're now in multiple species beyond E. coli, where we started. Now we're in Cerevisiae and difficult to work with gram-positives. And this enables new spaces that can be explored rapidly in a cost-effective manner. So because of the high probability of success, we're taking on projects that you wouldn't approach with traditional methods. And we're delivering for our partners, and as a result, we're growing and raising our Series B. So if any of you are interested in joining us in some way, please come find me after the talk. Thank you.
Thanks. Very interesting talk. So it seems like the way you get to those really diverse pools is by doing the fermentation to produce whatever product you're interested in all together in one tube. So if you're producing something, if you're trying to optimize the production of something that can diffuse out of the cell and into other cells, will you actually be able to identify the winners among them who are producing the most?
Yeah, very good point. So there's a couple of different ways we approach that. One, I mean, at the end of the day, when you want to look at the end product that is highly diffusible, you got to start separating the cells. So we have techniques for that using microencapsulation as well as plating. Sometimes often works and allows you to enrich. But then there's the other tack we sometimes take is engineer a sensor for a upstream metabolite that we can make sure it doesn't leave the cell, at least not quickly, and optimize for that, and then you push flux down the rest of the way. Okay, thank you.